Hi, everyone. I'm grateful for the opportunity to share my work with you. Thanks for the introduction, Dr. Gellenberg, and for the invitation. Um, I'll be introducing CellTune, which is a human-in-the-loop framework developed for efficient and accurate cell classification in multiplexed images, and this is also unpublished. Um, and as Dr. Van Valen mentioned, we're working also together to kind of use this tool, generate a lot of labels, and maybe have something more generalizable in the future as well. So I'm sure most of you are familiar with our typical computational analysis pipeline for multiplexed imaging, and the cell classification comes in after the segmentation. There are all kinds of methods that have been developed, gating, clustering, pixel clustering, different machine learning predictions. I show here an example of clustering, which is just still quite prevalent. And what I wanted to emphasize is the importance and motivation for this. Naturally, all of our downstream analyses are highly dependent on accurate cell classification. We haven't yet been able to say to what extent exactly. I'm sure it varies widely based on the biological question and the data, the statistical prevalence of the cell types and the microenvironments that are of interest. But our goal here is to approximate as best as possible human level accuracy, like manual labeling, which we found has been far superior than any of the available methods up till now. Although human labeling takes an inordinate amount of time, so we can't do that. But what makes cell classification so challenging? Unlike isolated cells in our tissues, we have cells that are densely packed and they're in 3D. We get a 2D slice, this image, and there's a lot of spillover of the signal and overlapping between neighboring cells. There are also cellular projections, which can go around neighbors. And of course, we always have some noise, and there are still some segmentation errors despite the big developments in that. All of these end up causing us to have many cells and many clusters of cells, which co-express multiple incompatible lineage proteins like CD20 for B cells and CD3 for T cells. So we get these clusters of cells which have both, and that's problematic for classifying them. Some recently published works have tried to address these issues in different ways. Some examples just to highlight that there's been some work done in this space. So Celesta and Stellar most recently, and ultimately, all of these require either removal of the spatial expression information that we see by eye, and or some clustering and gating, manual labeling and curation, which also takes a lot of time. For example, with pixel classification in Pixie, which itself requires clustering and some manual labeling of the pixel clusters, there's an additional step at the end where you need to cluster the cells. And then we kind of keep getting back to these issues. So our first question was, can we utilize the spatial information in the images we see very clearly? Our first try at that was to use a CNN, and this is a tool called CellCiter that was published in Nature Com this year. So we manually labeled a lot of cells and used an ensemble of CNNs to classify the cells along with confidence scores. And what we learned is that this really requires a lot of manually labeled training data, and it has very limited interpretability when we want to see what went wrong or why did it go wrong in making certain classifications. So this wasn't really the most efficient for us. And we asked, what is the most efficient way to kind of learn this? There have been a lot of these linear approaches. For example, you take a cell table, you do some clustering, then you need to identify what types of cells you have and label the clusters. Or you have some probabilistic model, which makes predictions, and then you use supervised learning with the manually labeled training data. But in all of these cases, we found that we needed to go and do a lot of manual curation on those labels, which takes us months and months of work. And in order to really minimize the amount of manual intervention, we turned to this human in the loop framework, also known as active learning, and found that we could use this efficiently for cell classification. The principles that really guided us were, as I said, to use the spatial features to be able to learn from human knowledge and to minimize the labeling efforts. Clustering and gating in theory would only minimize the labeling, and the supervised CNN did not minimize the labeling, but it allowed us to learn from what we labeled and from the images. The goal for CellTune was to be able to do all three. And so the workflow, we need to have some labels for training and features to learn from, as with any supervised machine learning. And then we train and predict. As with any active learning framework or human in the loop, you need to measure uncertainty somehow to allow you to sample new instances for the manual labeling. And then you need to be able to label them. And continuing in this loop until you're satisfied. In this case, we could even add features along with adding labels if we found that we're missing something in the data, and we could, I'll show how we could do that. But before you could begin with any of this, you need some initialization, some initial starting point of labels and features that kind of get this rolling. So what we did, we extracted spatial features from each cell, and in each marker, we took the segmentation that we had and we expanded it, and then we shrank it each time a few pixels, and we looked at also the wider environment around the cell, each time calculating the expression of each of the markers in these regions around or within the cell. And then we looked at for each cell what was the maximum expression of each marker by its neighboring cells. And this helped us capture even more spatial information, including those spillover effects where you have a really high expression of a marker on a neighbor and low expression on the self. And what's nice about this, the way we've set it up, is that it's flexible for any kind of input or feature. So we've had people creating regional masks in Elastic of things that they're interested in that come up morphologically, like crypts and villi in the gut, or for smooth muscle vasculature structures which express SMA, you could now differentiate them from myofibroblasts expressing SMA, and easily add that information in the form of an image. You could even add in something else, like anything you bring in, like a pixel classifier or other cellular features. And then we get this cell table with the cells and all the features that they have. But how do we initialize now some labels for them? We were inspired by a paper from 2015, Spitzer, where they do landmarking, which is really just using a simple high threshold where you're very confident about the cells being accurately labeled. So this ends up being around 10% of all of the cells for us, where we set a really high threshold on the different markers, and also that negative thresholds, like they shouldn't express other markers. So macrophages here, as an example, having really high CD68, and here's an image example of that cell, which is going to really pass this kind of threshold. It's like super clear what that is. And that's our starting point for training. So we train and we predict with gradient boosted decision tree models like XGBoost and CapBoost, because they're very, very fast and they're very good at learning from tabular data when you don't have hundreds of thousands of labels. So what we do, actually, we take two different models, we train them independently, and we get predictions from each one, and we compare them to each other. And in this way, we can measure uncertainty and where we should be labeling. So this is a well-known tactic called query by committee. So you take an ensemble and you look at where they're disagreeing with each other. So for here, we're just taking two models. We're selecting the cells where they disagree, um, and and this still ends up with you having a lot of cells in the beginning. So you need to then also sample from that subset of cells where they disagree what you're actually going to label in this loop, or this specific cycle. So yeah, the next question is like how do we actually sample the specific cells that we're going to label? And here we want to balance by default, just balance the agreement across different fields of view, different patients, different cell types, and we give some priority to rare cell types, which are often missed. And it's really flexible that the user could input any type of cell type or any type of confusion between different cell types that might be interesting for them. So if you're doing a project that's asking about macrophages and you really want to be like super accurate in your differentiation between types of macrophages, you could put that in as, giving priority to confusions between those. And then you get to the manual intervention part where a person has to actually sit and label the cells, and that's also not really trivial. So our question was, can we make this process faster as well? Because this was also taking a lot of time. And we teamed up with the Parker Institute, and specifically Rob here, who is the main developer of the Mantis viewer. And we added a bunch of features to this so that it enables the user to label cells really efficiently. And now when you hover over a cell, you have a legend which will show you a sorted list of the markers and their expression. And so when you're doing this in the context of CellTune, you have a cell who's predicted to be two different labels by the two different models that you need to label. So you actually get the two labels, and in this case there was spillover of CD4 onto a CD8 T cell. The CD8 is shown in green, and it's very clear to the user this is CD8 T, and they just adjust the population by clicking on the right, and so that made things a lot faster. And we also streamlined this by ordering the different cells that we're going to label by their confusions. So if we have CD8T and CD4T confusions, we order those all and look at them at the same time. So we just go through a bunch of those in a row and don't have to spend a lot of time changing which proteins are visible, which is another kind of thing that you spend a lot of time is like changing the markers that you need to see. And so that brings us to the next thing, which is we added these groups called channel mappings, where you can just click and define a group of markers that will come up together. And this saves also a lot of time. So in this case, if we start with some kind of overview mapping, we can see the general structure where the immune cells are, where the crypts are, and if we're in an immune region, we can put on the general immune markers and see, oh, this is a T cell, and then decide if it's a CD8, CD4. So we're able to do this really quickly. Those were schematics. This is the actual view in the software over here of like all of our tiled cells for one round, and you zoom in on a specific cell, and then it's easy to navigate to the next tile by just using like a keyboard shortcut. So you're zoomed in on this cell, you make your decision, and then in this case it was a CD4, and then you just go. It just takes you to the next tile with the keyboard. So that's the method. To summarize, just again, before moving to the results, the principles, were to use these spatial features learned from the human knowledge, minimize the labeling efforts. How we implemented it is extracting features from the images per marker, training pixel classifiers, using supervised machine learning, the human in the loop framework, filtering by this query by committee, and then also allowing the human to put in what's important to them. And to minimize efforts, we have all these nice features in Mantis. So for results, we had a manual labeler sit and label thousands of cells. So here I'm showing 3,000 cells in graft versus host disease. So we saw that the agreement between the predictions from a different person labeling with CellTune was 85%, which is actually pretty close to what we tend to get with different people. But when we actually look at the cells where they disagreed and have another person go over those and have to say whether, one is correct, the other, or both are okay because it's ambiguous. So we see that actually the CellTune predictions are better than just taking the one manual labeler in that case. And it gets a very high accuracy when we took a group of 3 manual labelers and saw, what's kind of the consensus between the 3 of them, whatever the majority vote. And CellTune was higher than the highest manual labeler, although it's not a huge jump, but the jump between kind of a perfect gating strategy or clustering strategy is huge, because those are in the 80% compared to 95% for this. So that's we've been really happy with that, and these are just it's even more when you look at the mean F1 score, which balances both between the precision and recall. And these are just specific cell types. So we see that for certain cell types, a lot of the pixel clustering gating methods do very poorly on the F1 score, but they're not a lot of cells. So we want to do well across cell types. This method is really efficient compared to labeling randomly or by field of view, in that our accuracy saturates faster with less cells labeled. And we've done this for a lot of cells across 5 data sets. Here it's showing 1.5 million cells, but we've actually added a few recently. And I'll just quickly, I have a few slides left, so we'll finish shortly. We have an example from lung cancer. We see, even where there's a lot of cells mixed together, we're getting good representation, at least, of the different types of cells. And we have some cell types that were unanticipated, which come up very fast when we're doing this, human in the loop approach, because they actually see the data. So we see cells like CD8 Tregs, which we weren't expecting, and in this case, she didn't put NKT cells into the initial list. So you see, different combinations of marker that you weren't expecting very fast when you're doing this. And I'm working with Donna, who's also joined on this Zoom, and we found that it's really highly interpretable. So you can see, because of these tree-based models, what they're doing and why, and it makes a lot of sense that it's looking at CD8 and it's looking also at the neighbor expression of CD8, and if the neighbor is high, then it's less likely to be that cell type. And when it's looking at Treg, it's looking smaller in the nucleus. That's this erode5 tag. And when we look at all of the importance across all of the cell types, we can see for some features like tryptase, it's really just helping us to do a single type of cell, which is mast cell. But for others like CD45, it's really giving us information about groups of different immune cells at the same time. And it's using these different types of features that we added in, which is not just the images. So we have like combinations of markers with this max tag, taking the max of several myeloid markers, and we have these colocalizations, which is computing where they're expressed in the same pixels. So if CD4T is like CD4 and CD3 in the same pixel colocalized. So in summary, we achieved a high human level accuracy for the cell type classification. It's really been a fraction of the time of what we've been spending before that. and those labeling features really helped improve our efficiency. And human in the loop has really been the thing that kind of pushed us to this next level, facilitating cell type discovery and also, allowing us to interpret what's going on and fix things flexibly. So yeah, so thank you to my lab and to Donna and to the labelers and everybody, Liat especially, and
Right.
the Parker Institute. Thanks.
Wonderful. Thank you so much, Yuval. This was a really great presentation.